so much for joining us today for our first Science Spotlight. Today, I'll be chatting with Miranda Byrne-Steele and Chelsea Berngruber from iRepertoire about how their team is responding to COVID-19. Chelsea, do you want to start by telling us a little bit about what iRepertoire does?
Hey, yeah, sure. So I'm Chelsea Berngruber. I'm one of the research and development scientists for iRepertoire and also the assay technical support specialist. At iRepertoire, we provide next generation sequencing of the immune repertoire. So we have both products and services for researchers and medical specialists who are in the research field where immunology is concerned. We have highly sensitive technologies that can go all the way down to a single cell level where we can sort out single immune cells and provide back the actual paired receptor chain data. And then we also have bulk technologies. Our newest technology is DAM-PCR, which has capabilities of analyzing all 7 immune chains in one single reaction. So alpha, beta, delta, gamma, Ig heavy, kappa, lambda, all in one single reaction to really get a fantastic picture of what's going on in an immune system.
And Miranda, can you talk a little bit about what iRepertoire is doing in response to the COVID-19 crisis?
Yes, so I'm Miranda Byrne-Steele. I'm the Director of R&D and Operations for iRepertoire, and we're doing several different things in terms of our response to the COVID-19 crisis. Scientifically speaking, we're collaborating with a lab in China that actually has the first 7-chain immune repertoire data that they generated using on our iReflex system. So we have been helping them with the analysis of that data so that they can look at several patients as they progress through the disease from their initiation into the hospital. 2 patients unfortunately went to the ICU, but all survived. And we're able to look at this 7-chain immune repertoire of these patients with time. So what we've been able to see is really fascinating, and that's going to be published. It was just submitted last week, so it should be published soon, hopefully. In terms of other initiatives that are more pertaining to the emergency, there are 2 areas where the response in the United States is lacking, is because we're lacking supplies in terms of swabbing patients so that we can detect where this disease is, but then also an overlooked need was RNA extraction kits. Everybody's focused on the detection kits, but there's upfront work that has to happen in order for you to be able to run these assays. And one of those critical pieces is extracting the RNA from a sample. So we're actually collaborating with another company, which we'll announce in a couple of weeks, where we're going to be running the validation protocol for their new RNA extraction kit. So they have an RNA extraction kit already on the market, but they've made a simplified, newer version with materials and products that they already have on the shelf, and they're reassembling it into a brand new kit that we will validate, because we have a lot of experience doing RNA extraction, since RNA is our template for our immune repertoire assays.
So I have 2 follow-up questions for people who aren't quite as familiar with immune repertoire sequencing. Can you talk a little bit about what 7-chain sequencing means and why that's important?
Oh, absolutely. So the immune receptor is, your immune system is basically your forefront of your environmental and genomic interaction. So it's actually rearranging in response to the environment. So when you encounter any pathogen, it leaves an imprint on your immune system. Normally, people would look at one chain at a time. So they would look at one subset, like T cells or B cells, and they would look at even just one chain of those, because that's what the technology allowed. And so we can kind of see by how that chain changes with time, the imprint, the environment is how it's impacting the immune system. With our technology, and you see this by sequencing these immune receptors, these little tags that are sticking on these immune cells that rearrange in response to the environmental challenge. So what we're doing is that we're taking not just T cells and not just B cells and looking at one chain, we're taking the immune system as a whole, trying to get a holistic view of what all the different immune cells are doing at the same time. And we're doing that not just in COVID-19, but also for all different types of diseases like arthritis or cancer, or really any disease has an immune component, and you can examine the immune system for health status, for seeing what the response is to a specific pathogen or even an internal stressor.
So returning to your comments about the RNA extraction kits, once you guys have validated these, do you have any idea how they'll be distributed or how other labs might get access to them?
And so once we announce that partnership with the company, that company will sell those RNA extraction kits worldwide. They already have extraction kits in the market, but they're back ordered. And so they need to come up rapidly with a newer extraction kit that they can use all the other materials that they have. And also that will allow them a more rapid turnaround to get another product out the door that people could order in these labs that are trying to do the detection. Because when you're trying to do millions of tests or scale up to millions of tests, that's a lot of materials that you need. And this will allow them to gain access, or allow other labs to gain access to those materials.
With regards to the 7-chain sequencing, once you've completed that for a patient that was positive for COVID-19, what do you do with that information? What's the next step?
Yeah, so we're doing a couple of things. So in the case of the Chinese data that we have, they collected the samples at various time points, so not just one static time point. So we can actually watch the immune system change as the patient recovers. So one thing that we noted is a high expansion of the B cell component RNA, and the T cell actually was quite disappeared at the very beginning time points at the height of the disease. And that T cell component started to come back at later diseases. So you can look at the fluctuation of the different immune chains with respect to each other, but then you can actually dive into the data and go look for very specific antibody signatures within that data. So we do an analysis, a network analysis on the B cell Ig heavy locus. So this is the B cells that are actually going to be the ones that initiate this response. And we can actually identify putative antigen-specific B cells directly from the immune repertoire data. But on the other side of that, we also have single cell technology. So immune repertoire, you're looking at a broader picture and you're doing additional bioinformatic analysis to tweeze out what the actual signature is. With the single cell technology, you can actually pull down antigen-specific B cells. And I think Chelsea can talk a little bit more about what our initiative is on the single cell. We're basically trying to enroll local patients with COVID-19 so that we can get access to the antigen-specific B cells. And this is like looking for those needles in a haystack, but those needles, those single B cells that are very antigen-specific, are a potential source of therapeutic antibodies. And I'll let Chelsea talk about that briefly.
Right. So we're kind of coming at this, like Miranda said, at a worldwide standpoint where we're trying to help validate these new RNA extraction kits that can be shipped out to any lab in the world. But on the local forefront, what we're hoping to do just at iRepertoire specifically is, we're hoping to find local patients that have tested positive for COVID-19 and take samples from them. And then, like Miranda was speaking about, we can do the single cell pull down and hopefully identify certain antibodies that are specific to this COVID-19 disease. And with this data, what we could do potentially is develop antibody-specific treatments, basically, which they're, I believe, neutralizing antibody treatments. So basically what would happen is this could be, even if you contracted the disease, this treatment could be used almost as a medicine to render the virus incapable of further attacking your cells, even if it's already been inside your body.
We didn't actually say where you guys are located, so maybe you could add that, Chelsea, and then have you gotten any samples yet?
We're located in Huntsville, Alabama, which is inside Madison County. It's about an hour north of Birmingham, hour-ish, hour and a half-ish. I don't there are positive cases in Madison County. I don't believe at this time we've taken any samples from any of the positive cases.
No, we haven't. We haven't identified a local donor as of yet.
Right.
And what do you think you need, or what could help with that step of identifying a first local donor?
We're working with several people who have contacts with the local hospital. We think that that's probably the best way if we can get an IRB approved study relatively quickly through the hospital. Once we get somebody on board who understands what we're trying to do, that would be a great step forward. They would just need to enroll the patient, draw the blood. We would go collect the blood, and we would do flow cytometry to identify those antigen-specific B cells, plate them as single cells in a plate, and using our iPair technology to co-amplify the heavy and the light chain pairs at the single cell level in the PCR plate. And then we have a way that we can actually screen those pretty rapidly using an in vitro transcription translation system. So we generate the protein small scale, and we do an ELISA assay to screen it. After that, we'll rely on partners to scale up the protein production and do the neutralizing assays. So we will need partnerships after we'll get the data to a certain point, but we're definitely looking to partner with people who are interested in developing therapeutic antibodies and have the capability of doing the neutralizing assays and scaling up the protein expression.
Excellent. I won't take too much more of your time, but I noticed Chelsea looks like she's sitting in her living room. Maybe you guys could just comment on how you're keeping things moving forward while still making sure to keep your own team safe.
Right. Yeah. So I'm at home now. I was actually at the lab earlier today, finished up what I needed to do, and just came on home rather than hanging around. Rufus has decided to move around every time I've needed to talk.
Let us see Rufus. He should. He needs to photobomb us.
Make an appearance. But basically, we're trying to keep things moving as smoothly as possible. We've kind of broken down into shift work. So we've divided up the teams into groups. Groups can sign up on a sheet when they need to go in, which labs they'll be in. And basically, we're trying to keep groups separate from each other because within each group is a backup for the other person. So basically, if group 1 gets compromised or someone gets sick and they can't come in, group 2 would be able to step in and keep things moving forward, because we do still have customer samples that we're processing. We're trying to, everyone's very understanding, but we're still trying to uphold our end and our basically get them the data that they've requested.
Excellent. Is there anything else that you guys want to add?
I don't have anything. Chelsea, do you want to say anything?
I don't think so. I think we covered most everything.
We covered everything that we're doing. Yeah.
Well, thanks so much for taking the time to chat with me today. And thanks to all of you who are watching for tuning in. Stay tuned for more videos in the future from the researchers that are on the front line in the COVID-19 crisis. Take care.
Thank you. Bye.